Yeah, welcome, Dr. Sekri. I can see you.
हाय
Dr.
डॉक्टर गोडसे, गुड टू सी यू.
Yes, yes.
एटलीस्ट हाँ, वेबिनार्स का ये तो फायदा कि एटलीस्ट यू कैन सी एटलीस्ट ऑनलाइन.
Good evening, doctors. Welcome to this webinar with respect to place of Ustekinumab in plaque psoriasis. I'll be speaking on the quality part. Dr. Rajiv will be talking about plaque psoriasis and its management and Ustekinumab place in plaque psoriasis therapy. And our moderator will be Professor Dr. Kiran, and our panelist will be Dr. Anil. So over to you, Dr. Kiran Gupse.
Yeah. So welcome to this World Psoriasis Day webinar. In fact, today there are so many activities of Psoriasis Day or by all over India by various companies and IDWL. So this is good. Something new has there, that is Ustekinumab, place of Ustekinumab in psoriasis. So this is a 1 hour webinar. We have very senior experienced consultant with us. So we have Dr. Rajiv Sekri, senior consultant dermatologist from Fortis Hospital, Noida, who will be speaking on Ustekinumab in psoriasis. And then Dr. Santosh Nair will speak about Usterirel, their world first biosimilar. So without wasting much ado, Dr. Rajiv Sekri, please, uh, stage is yours.
थैंक यू डॉक्टर गोडसे. आई शेयर माय स्क्रीन. So this is the molecule which I have been waiting for so many years. Now why? Because the convenience. Patient needs to visit the doctor only once in 3 months. And the patient is happy, we are happy that once in 3 months, and all those 3 months, the patient will stay comfortable and will say, thank you. Now psoriasis is working to his satisfaction. So I have been waiting for this and looking forward to very good results. I have already used it in two patients, but since it's a new molecule, so experience over a period of time, we will have it. So whatever we know is all from the published data. We all know that the biologics are the bioengineered monoclonal antibodies produced by recombinant DNA technology and are made by laboratory animals, mainly the mouse, against a target antigen, and are used to treat severe and refractory cases of immune mediated diseases. As I said, that this is a monoclonal antibody, and mouse is the main laboratory animal. So all that monoclonal antibody which we get will have some component of this mouse. If the component is up to 34%, then these are labeled as ZMAP. When it is up to 10%, it is labeled as ZUMAP. And when it is fully human, then it is labeled as UMAP. Now, what is the importance? As the murine component goes down, the immunogenicity of the biologic goes down. So this is very important. And we know that infliximab, I used first time infliximab in 2007. I have used it for many years. Now rituximab, which I am using in all those patients with immunobullous dermatosis, that is the drug of first choice for me to use rituximab for pemphigus or bullous pemphigoid. I have a large number of patients on this. I'm very happy. Then comes to the Zumab, Ixizumab, IL-17A blocker. Oh, I have used it in, I think, more than 100 patients so far. I'm very happy. So similarly, Omalizumab and IL-36 blocker, Spesolimab is being available in the Indian market, but I have not used it because it is just being introduced. And however, the other UMAPs, which is adalimumab, secukinumab, this is what we have used in a big way. And now ustekinumab is the molecule which is in my on my list, and I am going to use it in a big way. The advantage is that with ixekizumab, I have to use it say every month. Ustekinumab every month. And this I have to use once in 3 months. And the cost advantage, like this is very expensive, this is very expensive if you use it, and then this will be saving the cost to the patient and to us using it once in 3 months. We are happy. And we are talking about this, then we cannot forget about the fusion proteins are created artificially by recombinant DNA technology. We know that. And then the human dimeric fusion protein, the eternal cell, it has been used for the past so many years and still rheumatologists love it. But what we are going to today discuss is about the ustekinumab, and these are the biologics which we use in dermatology: TNF alpha blockers, IL 17A inhibitors, CD20 inhibitor, IL 36 inhibitor, IgE inhibitor, IL 4 inhibitor. Like dupilumab, they once approached me, but thereafter I have not got any inputs from them. But today our main talk is on the ustekinumab. So this is basically blocks the IL-12 and 23, because is a P40 subunit inhibitor and is used for psoriasis as a dermatologist, and psoriatic arthritis is the what rheumatologists are going to use. And then our co-specialist Crohn's disease and selective ulcerative colitis is something which is beyond our territory, but we are using it and we are going to use it in a big way in psoriasis. Like ixekizumab or secukinumab, this blocks only the interleukin 17A, and this is what the Th17 response that will produce the interleukin 17A, and this only binds to this. Similarly, TNF alpha blockers, they bind this and will inflammatory cycle. This is responsible for ongoing psoriasis. Whereas ustekinumab, since it blocks the interleukin 23, so this Th17 itself will producing 17A, 17F, IL-22. So it's not only the like ixekizumab or secukinumab, only 17A blocker. So this is going to help in reducing the pro-inflammatory cytokines, not only the 17A, F, and 22 as well, 26 as well. So this is going to show us the better results. This is very important. Now, second, we know that all those ixekizumab uh, or say secukinumab, they block only one cyto interleukin, whereas TNF alpha blockers only block only one. And what is special about ustekinumab that blocks IL-12 and 23? So this is something very specific. The reason is these three cytokines, they have P40 subunit, P40 subunit on it. So this ustekinumab basically binds to the P40 subunit. Since it binds to the P40 subunit and this subunit is present on interleukin 12, so that is blocked. So similarly, this P40 subunit is present on the IL-23, this is blocked. So both the IL-12 and 23 uh, uh, is blocked. So since both are blocked, so the inflammatory cycle will be brought under control in a better way. So this is very important to understand. And now, from the available published data, so I have chosen this to share with you. In this Phoenix 1, and it is being used as an induction dose of 45 milligram subcutaneous on the day one, and then after 1 month, and thereafter maintenance every 3 months. So if the person is 100 kg, 100 kgs under, that basically for most of us are going to use in day-to-day practice. So I'm not going to discuss about 90 milligram, but the 45 milligram. So what we have seen that when this is used for 12 weeks, day 1, then day 30, and then once in 3 months. So at 12 weeks, you see the PASI 75 is achieved in 67% patients. So this is the other study. In this is study also, it is achieved in 66.7%. So 67% patients, they are achieving the PASI 75 at 12 weeks. And when this molecule is continued at 28 weeks, you see the PASI 75 is 71%. 69%. So if you round it off, so we can say 65 to 70% of the patients will achieve PASI 75. Means more than two-thirds patients will be happy because the 5 has been achieved. And if you see the PASI 90, in about 41%, 42%, at 28 weeks, 49%, 44%. So we can say that about 40%, if you, I mean, average it out, you can say that 40% of the patients will achieve PASI 90, which is 90. PASI 90 is a great clearance to get the results in just 12 weeks and then maintain it at even 28 weeks. And you see the response, if if 100 patients they achieve PASI 75 and then they continue it for how much? 4.5 years. So this is American Academy of European Academy of Dermatology publication 2017. It says that even when you use it for 4.5 years, what you are maintaining from 100, still 80% of the patients will keep that PASI 75 score. Now, it's a great thing. How 4.5 years? It's a big data that after 4.5 years, still 80% patients will maintain the PASI 75, which is a very good result. And similarly, PASI 90, about how much? If you say PASI 100, one third, 31% patient, 30% patient. So PASI 100 will be maintained for 4.5 years of the data. We don't have a data beyond this 4.5 years so far available. I have looked into all the literature in American Academy, European Academy, and all other publications, but I could not get a data beyond that. So this is a very good achievement. So we are looking forward to see our patient to fall in this category. And second, some very important thing to share is that, as I said, that this is blocking this IL-17 pathway. So what is happening? It is not only this 17A, 17F, it is the IL-22 also is blocked. So IL-22 is responsible for epidermal hyperplasia and acanthosis. Hence, this works very well in palmoplantar psoriasis. We know that palmoplantar psoriasis is very tough to treat, and we have to use many times the acitretin along with other antiseptic molecules to get the results in palmoplantar psoriasis. And now this is in a very good. I'm looking forward to treat patients of the palmoplantar psoriasis with this. And then off-label use of Ustekinumab, hidradenitis suppurativa, pyoderma gangrenosum, areata, lupus erythematosus, PRP, Behçet's disease. Now, these are basically case reports at random, uh, and but we need to have in a proper. Dr. Godse can definitely take up some research on this, these patients. But these are the some papers published, and based on that, this is the off-label use for the ustekinumab. And now the concern is the use in pregnancy. All the biologics, we know that there is no No biologic says that, oh, fine, fine, you can use it in pregnancy. But we know that in the third trimester, so this is an immunoglobulin, will pass on to the baby through the placenta. So it is recommended that all the biologics should not be used beyond 20 weeks of the pregnancy. But there is no, I mean, official permissions. They always say, please discuss and share with the patient and think about it. And with the breastfeeding also the same story falls. But I understand that if a small amount of the molecule passes in the breast, this is in a IgG, it's in a immunoglobulin, it's in a protein. The moment it goes to the stomach, then there is an immunoglobulin, and hence the systemic absorption is negligible. So to my mind, we shouldn't be too much afraid. Though the literature says don't use in or be careful when the lady is breastfeeding. And then the latent TB. Yes, we know that this is the IL-12 blocker as well. And since it is going to block the IL-12, so TNF alpha blockade is going to happen. And we know that the TNF alpha acts as a primary fighter for the tubercle bacillus. Now, if that tuberculosis or the tubercle bacillus is present in the patient and it is in the latent stage, so this can be reactivated. So it's very important that the patient should be worked up for the latent tuberculosis. So I advise all patients to go through the Montu X-ray chest and IGRA TB Gold to be very sure that there is no latent tuberculosis. If the things come positive, then I send my patient to the pulmonologist and request them to give the green signal and guide me how should I go about it. And then again, very important. Now, this molecule can worsen atopic dermatitis in patients with atopic diathesis because it can reactivate Th2 response. So the patients who have psoriasis and atopic dermatitis. Now, this is what is called spongiotic psoriasis. The patients who have spongiotic psoriasis, they are the psoriasis with in the background atopic diathesis. I think we should be careful in prescribing this molecule. And about the immunogenicity, the anti-drug antibody formation is seen up to 12.4% of the patients. It's a large number. And it is important to make a point that the majority of these anti-drug antibodies are drug over a period of time. What will happen? This is what I have observed with all those TNF alpha blockers, even secukinumab, or even ixekizumab. Like with
I think
Dr. Sekhri, I think you need to share
yeah,
your screen again.
yeah, Dr. Rajiv, we have
yes.
lost the screen. Yeah, we have lost the screen. Can you please share the screen again?
Okay, now you are not seeing the screen.
No,
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 doctor. No.
Okay, I do it again.
Yeah.
Now
Now
you can
we
see
can see.
it.
Yes, yes, doctor, we can see.
So have you seen this pregnancy point? No?
you start with
Hello?
atopic dermatitis. Atopic dermatitis.
Hello?
We were in the
Hello?
immunogenicity. Yeah.
Yeah.
Okay. Okay. So you have okay. So you have seen this atopic dermatitis. Okay, fine. Now, as far as the immunogenicity is concerned, up to 12.4% patients have developed anti-drug antibodies, and the majority are drug neutralizing antibodies. This means over a period of time, there is going to develop the biologic fatigue, and we need to remember that this is an immunoglobulin, and all the body this as a foreign protein and going to develop the anti-drug antibody. And this is seen with all the biologics, even secukinumab, ixekizumab, or adalimumab, or infliximab, all develop that anti-drug antibody. Over a period of time, usually over a period of 3 to 4 years, biologic fatigue develops. Even with ixekizumab, which I'm using for so many years now, and the biologic fatigue has developed. So this is what is expected. So if develops, nothing to panic. But in 12% patients, this happens. But rest of the patients, they will continue to respond. If somebody develops a biologic fatigue, yes, we can always switch over to another molecule. And then the patients can have other side effects like hypersensitivities and LE-like phenomena. Now, to prevent this biologic fatigue, we can combine the molecule with low methotrexate. So we can give ustekinumab plus methotrexate 10 milligram to prevent. So advantage is it will reduce the immunogenicity plus it will give us the add-on benefit as an antibiotic treatment. So it's a win-win situation. So this can be combined with the etanercept as well. There are few papers published a combination with etanercept, which I think I will not do that. And methotrexate, yes, I will do that. And acitretin, yes, I have no problem. I can combine it with palmoplantar psoriasis patients. And thank you very much. Any questions, discussions? We can go
Thank
ahead.
you, Dr. Rajiv, for wonderful talk. We'll have a question answer after Dr. Santosh Nair talk. So I request Dr. Santosh Nair, who
Yeah.
is heading medical affairs from Reliance, will tell us about the Ustekinumab, the first biosimilar of Ustekinumab, which is launched now in India. We did trial of this 4 years back. So let us hear from what are the results. Dr. Nair, over to you.
Yeah. Dr. Rajiv, can you stop sharing the slide so that I can share mine?
So I have stopped sharing.
Yeah, you need to stop sharing so that I can share mine. Yeah, that slide.
Yeah, I have stopped sharing.
Yeah.
Okay. Yeah, we can see the screen.
Yeah, I will just put it in the slide mode. Good evening, doctors. So I'll just brief with respect to our Ustekinumab by the brand name Ustekinumab, which is basically world's first biosimilar, which we have launched a month ago. Basically, to understand, we at Reliance Life Sciences, we are the most diverse integrated biotech firm and globally considered number one in terms of biosimilars, as we have 23 biosimilars in the market. We are also leaders in the plasma proteins in Asia and pioneers in regenerative medicine in India, as well as we are into innovative technology with respect to gene therapy, vaccines, mRNA products, fusion proteins, and microbiomes. And global presence, as I just conveyed, that we are having our footprints in 101 countries across the globe. And coming back to the innovative technology from the innovator, as well as since we are a biosimilar, we follow the same protocol in terms of the production of this product. As Dr. Rajiv has already mentioned, the commercial production is done in mouse SP2O cell lines. And because we replace the immunoglobulin genes of the mouse with the human immunoglobulin genes, and wherein we develop this biosimilar, that is Ustekinumab. And as Dr. Rajiv has already dealt upon the unique mechanism of action, it binds to two different cytokines, IL-12 and IL-23, Basically, it binds to the common subunit, that is P40, which is present in both IL-12 and 23. And therefore, this unique mechanism of action also prevents the release of other interleukins, including interleukin 17 and other interleukins, as Dr. Rajiv had already mentioned. So these are the advantages because it has got a unique mechanism because it binds to two prime cytokines, 12 and 23, and the half-life is 20 to 32 days, and therefore the infrequent use of ustekinumab every 12 weeks is favorable because the patient compliance is very much there, as Dr. Rajiv told, that it is very, very easy for the patient to continue. And in fact, compared to other monoclonal antibodies used to treat plaque psoriasis like secukinumab as well as ixekizumab, where they have to take every month the injection and the infrequent administration, and it has got the subcutaneous route of administration. So the patients have a high medication adherence rate because of the compliance, because they need not come every month, and basically it is a convenient option for the long-term maintenance. And it is easier to use it in obese patients as dose can be adjusted. Two strengths available, as we all know: 45 mg for patients below 100 kilograms of body weight, and 90 mg for patients above 100 milligram, above 100 kilograms of body weight. But mostly we are using 45 mg because most of the patients are below 100 kilograms of body weight. It is very, very effective in terms of rapid disease control as well as in terms of intermittent treatment, and being able to use it in comorbidities as a first choice in demyelinating diseases. There are papers where even it has been used in multiple sclerosis, it being a demyelinating diseases with good results, but being an off-label indications. Not much paper published, also used in lupus erythematosus, and also even in case of a heart failure patient. So basically, we need to consider this important. One second. Basically, we need to consider when we come to the quality of the product, when you are creating a biosimilar in the laboratory, which is 100% similar with that of the innovator, there are many, many important points we need to consider, which are very, very critical. It all starts with the cell line source. And as doctors, we know that Lonza and Horizon, these are the two major cell line suppliers in the global market, right? So Lonza and Horizon is supplying cell lines to all major companies, all who are involved in the manufacturing of biosimilar, because they provide highly efficient bacterial or mammalian expression system cell lines. And they also provide special cell lines for direct expression of folded protein, which is mostly preferable by most of the companies. So why a folded protein it automatically gives means the interference of the chemicals are reduced, because otherwise if you want to fold the protein, which is part of the manufacturing, you need to add certain chemicals which may interfere with the patient's treatment. So when you get a direct folded protein during the manufacture, it will be easing out the use of all these chemicals. So that is preferable. And the advantages of these special cell lines are there. They are high producing cell lines, and they're consistent in terms of product concentration, growth, and product characteristics, which is very important. And chemically defined animal components, they are not present in such special cell lines. And then we also look into single cell cloning, because this is a process wherein a cell line clone is developed from a single cell, and they are developed into the master cell bank for further manufacturing. So we use fluorescence activated cell sorting, which is performed to deposit a single cell into a vessel of a tissue or a culture plate. And each cell will be injected with the ustekinumab gene, and under standard conditions of temperature, pH. Several colonies are analyzed, and the top clone with the best protein. That means the best protein, I mean to say, is in terms of the structure, fully developed structure similar to that of the innovator, as well as the amino acid sequence, the electrical charges, everything, the molecular weight, everything has to be exactly 100 on 100 with respect to that of the innovator. So the best one will be selected for further production. This will help in the consistency of the quality, batch to batch consistency, and uniform across all the batches. So we create the master cell bank characterization because we use the best single clone of the cell line, which is basically multiplied to form the master cell bank. And then, routine periodic check of the master cell bank is done because there are certain guidelines like the ICH-Q5D guidelines, which talk about, how to maintain the master cell bank. So you look at the purity and you look at the viral contamination, etc. And because this is also very, very important, we need to follow a two-tier cell banking. We have a robust cell line source storage system. We have huge, basically, containers where we develop this product. And to ensure the preservation of the lineage of the original cell line, two-tier cell banking approach is always recommended globally. And this is characterized, basically, we create a master cell bank, which is characterized extensively, and from the master cell bank, we create a working cell bank, which is WCB, which is then characterized prior to use in manufacturing. So from the working cell bank, we remove the cells which have been, obtained from the single cell clone, and we continue to produce batch after batch with the same consistency. And most important, the use of a disposable bioreactor is very, very key. Because if you use a stainless, steel bioreactors, the chances of cross contamination is there, which can carry forward, which is one of the highest risks. And it is one of reasons for infusion reactions. And because there is insufficient cleaning and sterilization of the equipment between batches. So one batch is over, and the second batch, when it is taken up, you need to have a proper sterilization and cleaning technique, which sometimes, if it is missed, it can lead to contamination of the product, which is finally, when it is in the form of a PFS, and this can, once injected in the patient, can lead to infusion reactions. So what we use is a single-use bioreactor, which is also called as the disposable bioreactors. And this single-use bioreactors offers distinct advantages over standard stainless steel bioreactors because they do not get there is no need for cleaning, because once you finish one batch, that bioreactor is disposed of, right? And when the new batch is taken, a new bioreactor is used. So does not involve cleaning, new batch, new vessel concept. So every batch there is a bioreactor, and after every batch is completed, that bioreactor is basically disposed and a new bioreactor is taken up. And this will eliminate the risk for cross contamination as well as the risk to the patients in terms of any adverse drug reaction like infusion reaction or injection site reactions. So this is what basically our regulators look at. They look at the protein fingerprint because this is very, very important. They want to see that your product is biosimilar, means it is the exact copy of the innovator. So whatever the innovator has designed the structure of the drug in terms of the molecular weight, the molecular size, the electrical charges, the amino acid sequence, and the IgG structure, right? So they look at the protein structure, they look at the drug related, they look at the potency, post-translational modifications, general methods, whatever methods you're using to look at endotoxin, sterility, etc., and the process-related impurities. Because you're using cell lines, there is bound to have some impurities from the host cell lines, which has to be eliminated by using various high-end chromatography techniques. So this is what is basically important for your biosimilar to get approved from our Indian regulators. There are panel of doctors, which includes the the expert committees, and they look at all these aspects and all this testing before passing out your product as fit to be used, and then it gets the license and it is coming in the market and to be used in the patients. So a short glimpse quickly into the physicochemical and the biological characterization, what we do. So you can look here. We do 22 orthogonal testing. Orthogonal testing means one single parameter we test by using 4 to 5 different methodologies. The answer has to be the same. So you look at each parameter in terms of the primary structure, the free cysteine content, overall charge, purity, the secondary and the tertiary structure, the process control. So by this way, we are doing a 22 orthogonal test to prove that our product is exactly a copy or a biosimilar to the innovator, which is there internationally. And this is one of the example of purity by SEHPLC, or size exclusion high performance liquid chromatography, which is a high end chromatography technique which will purify the product once it is passed through this column of the chromatography, and it will trap all those unwanted proteins which has been generated along with the drug, for example the host cell DNA or protein fragments, or part of the cell line. So all this will be trapped and cleared out and purified, and you can see that basically you can see the batches. You can see on the right hand side one batch of Stellara, which is the innovator, and four batches of our Ustekinumab. We have done the purity, and you can see basically the graphs. You can see they are touching with each other, and which clearly mentions that this product is as pure as that of the innovator, basically. So you can look at this one, and you can also look at that we also use the spectroscopy, basically can see that we need to analyze the biosimilars high order structure, looking at the glycosylation, protein concentration, and what is the excipient quantity. So this technique helps us to understand. So this is also basically compared with that of the innovator versus four batches of Ustekinumab. And the most important is the biologic assays, because it is very, very key to understand the mechanism of action. So the bioassays basically the bioassays measures the biological activity of a test substance. For example, here it is the ustekinumab or ustekinumab, and it is fundamental in establishing the efficacy of a drug as the assays mimic. So what mechanism of action is done inside the body that is mimicked, and the biological assay proves that ustekinumab manufactured by Reliance Life Sciences basically absolutely blocks the P40 subunits of IL-12 and 23, thereby preventing the binding of these interleukins to its respective receptors. So it is very, very significant in demonstrating the biosimilarity with that of an innovator product, because this is the key assay which the drug regulators need to understand, because your product is definitely blocking IL-12 and 23 like that of the innovator. So this is a key assay, and this depicts the actual mechanism of action, even though the assay duration is very long. So critical tier 1 assay is used currently to prove that it is absolutely same as that of the innovator in terms of its binding to IL-12 and 23. So this is the IL-12 signaling inhibition assay. Ustekinumab or ustekinumab inhibits 12 and 23 binding to the IL-12 receptor, beta-1 receptor, and prevents the dimerization. And it is detected as a decrease in luminescence signal compared to innovator as dose-dependent response. So you look at the luminescence signal, which is basically a technique. Decrease in the luminescence signal, which clearly talks about the 100% binding of ustekinumab, binding to P40 subunit, which is common in 12 and 23. So this is, I'm not going to talk much about the clinical trial, but I'll just give an overview that we had done a multi-center randomized double-blind two-arm trial. We looked at the efficacy, safety, immunogenicity, and pharmacokinetics of ustekinumab compared with that of the innovator in moderate to severe plaque psoriasis. And this trial went for 52 weeks or 1 year. And we compared ustekinumab with Stellara, and the sample size was 220 patients, and we did this in 15 sites across India. So this was the study conclusion. The comparable efficacy was seen, the comparable efficacy profile to innovator as evident from the efficacy variables which were analyzed. We looked at various endpoints, which includes PASI 50, PASI 75, 90, 100 response rate, the physician's global assessment score, and the change in dermatology life quality index score, as well as the short factor 36, which were all very, very important parameters when the innovator has done all these parameters. So since we are a biosimilar, we need to look at all these endpoints, including the efficacy profile. And the safety profile was in line with known safety profile of ustekinumab, without any major safety concerns. None of the patients in ustekinumab group developed anti-drug antibody antibodies or neutralizing antibodies, or there was no immunogenicity in any of these patients who took ustekinumab, and none of the patients left the trial. They were there till the end of 52 weeks. So immunogenicity was negative in all samples, and single and multiple dose pharmacokinetics also showed comparability. So in conclusion, doctors, Ustekinumab, as Dr. Godse and Dr. Raju had already mentioned, it is the first approved global biosimilar. So nobody has manufactured globally. We at Reliance Life Sciences, we have manufactured this first globally approved biosimilar Ustekinumab, and we followed the protocols of single cell cloning, which is very important, the master cell bank, as well as the working cell bank, the master cell bank characterization. We also followed the two-tier cell banking, the disposable bioreactors, which is very important, so there is no cross contamination. Chances of infusion or injection site reactions are not there, and there is extensive characterization in terms of the product structure and protein, the amino acid sequence compared to that of the innovator. All the batches released are tested against very, very, very stringent true cell-based bioassay. As we have already seen that bioassay is a very, very key important test, basically to understand the mechanism of action and the binding of ustekinumab to P40 subunits of 12 and 23. So depicting the correct MOA. So you can see on the right hand side, doctor, right from interferon alpha 2b, we have now created 23 biosimilars in the market for various specialties, and many more in the pipeline to come. And we will see many more drugs in the market. Especially, we are going to focus more on dermatology and other branches of medicine. So that's what I wanted to talk about in terms of the quality.
Yeah, thank you, Dr. Nair, for that wonderful overview. Although we could not understand much of the technical terms as we are clinical dermatologists, but impressive, and we have been using your Reliance Omalizumab and other products, so we know, and this property happens to be very near to Navi Mumbai, where I stay. So,
Absolutely.
well, when a new biology come, obviously all dermatologists are excited. Something new has come. So, what are the advantages? We look at it, how its efficacy, safety, and comparability. That is what we see. So, Dr. Anchala, when it comes to you, the new biologic, what is your reaction and how do you look this Ustekinumab in your practice? Unmute, Dr. Anchala, unmute yourself.
Yeah, Dr. Anchala, you can unmute, please.
Yeah, the important thing which I noticed from the doctor, the one the previous lecture is that the bioreactors usually they are not disposable. Most of the times they are stainless steel bioreactors which are not disposable. But this is a disposable bioreactor. That is a really a good thing which I noticed because I had been there to the place where the biologicals are being manufactured. That's why I know the huge bioreactors and all those things. That is a good thing. Coming to Ustekinumab, of course, as the previous speaker is pointing out, we are waiting for that Ustekinumab since long time because it is easy to use 0, 4 and every 3 months. And then, of course, the side effects profile is not that very serious. Side effect profile is not there. And then convenient to use, but about the efficacy, but probably it takes a little bit slow time to respond. Probably it takes 4 weeks to 12, 8 weeks, something like that. But we are trying. We will try our best to see where it is placed. Once you get the results, then only we'll be able to tell you because real world experience is more important than the other these things. So of course there are quite a good number of real world experiences. Studies have been done from 6 years, 9 years, 7 years, and all those things. But in India, we have to see what are the real world experiences.
Yeah. So again, as I understand, this is not as strong as Ustekinumab, as we understand. It is not where you get PASI 100 in 2 weeks or 4 weeks. So this is slow and steady wins the race. That is what it is. And PASI 75 is the benchmark for most of the things. Dr. Rajiv, how do you place this molecule vis-a-vis others? See, against all safety, efficacy, availability, and economy, all the 4 perspectives.
Economy, it is the most compared to ixekizumab. Compared to adalimumab, this is going to be the most cost effective. And the more the affordability, the more the patients they use, and we are happy as the doctors. Now, coming to the side effects profile, definitely using the TNF alpha blocker alone is going to have more side effects. Practically, the reactivation of the tuberculosis, which is the major concern, if I am concerned, because in India we live in a tuberculosis prone area. Now, as far as the Ustekinumab publications are there, I need to understand that all those publications are from the West, where the tuberculosis is not something present in the society. So the publication says, "अरे यार सब ठीक है, सब चंगा है. There is no case report and yes, no reported." That's fine. First, it is the number of patients you have for the reports. Now we are living in a society where everybody spits, थूकता है जगह जगह पे. So we are the people who are going to see the relapses, recurrences of the tuberculosis reactivation. So I have made a point that we have to be careful. So if there is an IGRA positive, then we must take the pulmonologist as a team member to go about with the treatment protocol. Otherwise, there can be legal issues, right? So this is one important. Now coming to the other side effects. Now other side effects like other biologics, भाई थोड़ा सा URC हो गया, IL-17 block है, तो we become more prone to say viral infections or the fungus infections. So these are the with all the medicines we use, these are the chota side effects which if happen in some patient and then they are all reversible. So we shouldn't be much worried about. But the only since practicing in India, tuberculosis, we have to be more careful.
Yeah,
Baki koi, I'm not afraid of much of their side effects.
yeah. Dr.
Dr.
Nair,
Godse, you
actually,
want to add?
yeah, yeah. Basically, see, they have yet to publish the trial which we did, 220 patients,
Yes,
out of which
yes,
we had few patients. So,
yes.
what are the preliminary findings? If you can share, although it is not yet published, but you can share the preliminary findings. Anybody had got tuberculosis or anything, any major side effects?
No, basically, we looked at tuberculosis, so there was basically no tuberculosis there. And but immunogenicity was found in two Stellara patients, right? So Stellara definitely showed up a couple of immunogenicity, but there was no immunogenicity seen in Ustekinumab. And basically, the known side effects definitely yes, some of them with infusion reactions or injection site reactions, because it is already published that at least 1 to 2% of the patients may have infusion site reactions. Apart from that, no deaths occurred, and absolutely there were no other serious adverse events wherein we need to stop the drug, doctor.
Yes, yes. In fact, there are some questions from the panelists. There is one question.
Dr. Nair, something I, Dr. Nair, Dr.
Yes,
Nair,
yes,
I have a
doctor.
question
Yes,
now.
yes, doctor.
When you enroll the patient, when you enroll the patients for this study, then you
Hmm.
make a point that anybody who is having IGRA positive or TB
Hmm.
Gold positive is not taken into the study,
Hmm.
right?
Yes,
So, the results you can't generalize that.
right.
Okay, I'm sorry, but this is as a doctor, as a professional, I
Hmm.
have to make a point. Am I right,
Hmm.
Dr. Godse?
See, Dr. Rajiv, all the trials we
Yeah,
do we do IGRA for all, whether it's
IGRA.
Omar
Yes, yeah,
trial
yeah,
or any trial they do, it is mandatory. So, but
correct.
still, patient can develop tuberculosis
Right,
after this thing. Like somebody,
right.
in fact, one of the biologics patient developed tuberculosis lymphadenitis after 6 to 8 months. So, at the time of enrollment,
Yes,
we want a clean person. It's
yes.
like a military recruitment. We want the best of best.
So
So,
that inclusion and exclusion criteria is there for all
yeah, yeah.
these
So,
patients,
okay, so
doctor.
we
Yeah,
do. Obviously,
Dr. Nair, Dr. Nair,
Yeah,
can I add one thing?
yeah, please, Dr. Prasad.
Yeah, good evening,
That's what I
doctors.
said.
Yeah, good evening, doctors. This is Dr. Prasad Apsangikar. I am the overall head for medical in Reliance. Sorry to not have met you because Dr. Nair is taking care of all these products for you, and he's absolutely in touch with you. So, in terms of the eligibility for TB screening criteria, no history of latent or active TB, no signs or symptoms suggestive active TB, medical history, physical examination, no have no recent close contact with a person, uh, have no evidence of latent TB based on tuberculin testing, quantiferon TB, gold test, or other tuberculosis screening tests performed during the screening. Have no evidence of current active or old inactive TB based on the chest radiography. These were the TB inclusion criteria which were taken care of, sir.
Yeah, thank you, doctor. In fact, obviously, trial we do this thing.
Yeah.
One interesting point about this molecule is it is also can be used in Crohn's disease and IBD, inflammatory
Ulcerative colitis also. Yeah.
quality also. That's the advantage because IL-17 blockers generally we can't use in this thing. So typically we are worried, those patients, those who got constipation, diarrhea, some blood in the, we are worried whether it's a Crohn's or something else and we need to reverse. So this patient here, we don't really worry about the inflammatory bowel disease of Crohn's or ulcerative colitis. Dr. Anchala, how is it an advantage for you when you,
Definitely,
same thing is,
definitely that Kiran got said because it is useful for ulcerative colitis patients and Crohn's disease patients. Definitely, that scores the point over there. But the thing is the sustained response, even after 78 weeks of the therapy also, at least the maintenance of PASI 90, PASI 75, maybe around 30-40% PASI 90 and around 60% PASI 75 is really good, even after quite a number of years. That is the plus point here. And
yeah,
the only thing is anti-drug antibodies. That is a minus point because around 10% it is seen. That is another one, and they may lose their efficacy also because these anti-drug antibodies are definitely they're making the drug to lose their efficacy, not like other antibodies. That is one drawback with this. And then the patient, we have to tell the patient that you have to wait for the response. Unlike IL-17 inhibitors, you
yeah.
have to tell the patient you have to wait at least for 8 to 12 weeks, then only you will get response. Because when I gave to one patient and then he came for the second injection, the fourth week, he was telling, sir, response is not that much. I'm getting only 30%, 40% response. I told him to wait, take another injection and come back for the third injection. Then you know how much response you get. So the second injection recently we have given, so we have to wait to see the response.
Yeah, yeah. So response takes time, Dr. Rajiv. Yes.
How about hepatitis B and C, sir?
That is a routine for everybody. We have to do it actually.
Now, if the patient has, say, hepatitis B or hepatitis C, how you will go about? You will say no, or you will take some,
No,
say,
when you are
some.
when you are taking the help of a
Yeah.
a pulmonologist, why don't you take the help of a gastroenterologist? He will be there to help you. If he wants to give any extra investigations, he can ask for it, or he can give the antiviral therapy, and then we can start.
A viral load they do actually to find out the virus. Sometimes they start lamivudine. Basically, we give it to a gastroenterologist. Let them decide. If they give green signal, we give. Otherwise, you don't give. For most of the hepatitis. Dr. Nair,
Yes.
yes, anything about hepatitis B, C?
No, none of them seen in this completion of the trial. So we could not see anybody getting hepatitis B or C or even HIV. So we had screened all these patients and none of them developed, doctor.
Did you see any development of atopic dermatitis or exfoliative dermatitis or something like that in those patients where the trials have gone taken?
No, that is not reported in the trial. None of them had any atopic dermatitis, doctor. So we only looked at all the routine, as Dr. Prasad had mentioned, that inclusion and exclusion criteria was very important in screening for all these patients. And then post clinical trial also, during that follow-up also, we looked at all these parameters and we could not find any serious adverse event or anything related to atopic dermatitis or apart from that, hepatitis B. Yeah.
Yeah, but by taking the clue of this, it is important that those who have got high IG levels in 2000, 3000, 4000, you have to be extra careful. Because this molecule is can precipitate atopic dermatitis. So typically,
Right.
IgE also can be done.
Mhm.
So patients, just to understand those who have got atopic tendencies, we can don't give this molecule because it can worsen atopic. That is one thing we can take a
So you
lesson.
mean to say that it is better to do IG levels also in those
Yeah,
patients?
exactly. So those who have atopic tendency, if IG
Mhm.
levels is very high, it's better that you can give some other molecule and not this
That
molecule.
is a
That
good
is
point to
my
be taken.
initial
That's a good
presumption.
point.
Obviously, time will tell us.
Yeah.
Mhm.
Right,
even
right.
And
even for hepatitis B or C, I think if the patient is having the history or contact, then I think antiviral prophylaxis is also there in the guidelines before you start ustekinumab. Am I right, doctor?
Yeah, I think they are there.
one of the article, I think, ixekizumab induced Crohn's disease or ulcerative colitis
Yes,
with
yes.
plaque psoriasis. Patient was given with ustekinumab. They had a very good result.
Yeah.
So this is also
Exactly.
being
In fact,
published.
there is a publication that patient developed Crohn's, so he was given Ustekinumab, which
Correct.
took care of both the
Both.
Crohn's as well as this thing. So
So
that is
that was a dual advantage of the drug. Yeah,
so one drug and two diseases in same
exactly.
patient is something unique. There is one question from Dr. Sachin iPhone, that how do you counsel the patient and how long the treatment should be taken? Now, please understand that all the biologics do not cure the disease. They give better quality of life and long remission. So we can never say that I can cure your disease, but give a long-term disease-free life, and it is reasonably economical. That is what I can see. Yeah,
So
now
better quality of life. Better
yeah,
quality of life.
Yeah, they are meant for lifelong use. That is one thing. The patient can take it as long as he can afford to take it.
exactly.
Absolutely.
Then we can change. Dr. Sapna has asked, sir, if the patient is already on other immunosuppressant like methotrexate, do you need an overlap period or we need to stop? Dr. Rajiv, we generally with these biologics we do give methotrexate to prevent anti-drug antibody. That is our thing, our Indian jugaad, so that the action of your biologics goes on. So should you take wait and something, or you can continue and add to it, Dr. Rajiv?
I'm happy to add methotrexate 10 milligram, which is a very low dose with all the biologics.
True, true. We do that. In fact, typically in a private setting where patient has to use that molecule, and
In that
obviously
case,
that is the
Dr. Kiran Gad said, then how do you know that this Ustekinumab is working or methotrexate is working? I don't agree with that actually.
nine.
Suppose the patient is not responding to Ustekinumab, maybe if you want to add methotrexate, okay, you add. But adding from the beginning methotrexate plus Ustekinumab is a wrong idea. I don't think it's a good idea.
Okay.
Then which one is working? How do you know which one is working?
Yeah. Now see, the purpose of adding methotrexate is to prevent anti-drug antibodies and give added response action. Obviously, this drug being a weak drug, it is not a strong drug. So some people, obviously in trial we have done nothing like this, but this is a Indian jugaad just to add the action, add the benefit of the drug for longer time. But anyway, it is individual opinion. Nothing personal on this. There is one more question: can we give cyclosporine along with, again the same thing? So can you give cyclosporine, Dr. Rajiv?
No, I will not.
Yeah. So cyclosporine is a very potent immunosuppressant, and I don't think we should give two immunosuppressant, because even this is to some extent immunosuppressant. And yeah, now this is a question about the cost of this biosimilar. Dr.
Yeah,
Nair, give us rough
yeah.
idea, because many people are interested to know how much is the cost approximately.
So I will ask Pankaj because he's looking into the price part. Pankaj, are you there? If
Yeah.
you can just brief upon the cost of 45 milligrams pre-filled syringe.
Yeah, good evening, all doctors. Regarding this question, 45, we have two strength 45 mg and 90 mg. 45 mg costing is 50,000 is the MRP right now, and 90 mg having the 1 lakh is the MRP.
This is including the GST, right?
Yeah.
And there is a scheme also. No? You have got some 3 or plus.
Yes.
So ultimate
Yes.
it cost comes down.
Yes.
Yes. If the patient takes, because in a year he needs 6 PFS. So if he purchases 4 PFS, he gets 2 of them free.
Yeah. So it comes at the end. Total cost comes down with that.
Much economic benefit compared to Secukinumab and Ixekizumab, because that is a more costlier one.
So it is 3 plus 1. This is what it is?
It is 4 injections they have to purchase, 2 will be given free.
No,
So total 6.
No,
Plus
no,
one.
sir.
It is two-
No,
two plus
Dr.
one.
Nair, no, sir.
Yeah.
No,
Two plus
sir.
one.
To 4 plus
Wait. Kadu, बोलिए.
Yeah, बोलिए Pankaj.
4 plus 2 is not there, sir. 3 plus
Acha.
1 is there.
Achha, it is 3 plus 1.
Exactly.
Okay, fine.
3
Okay.
plus.
Yeah, sorry, Pankaj. It is 3 plus 1. Sorry.
So that is still less. The cost approximately, you got the idea. Obviously, and then somebody is asking, is it better than Secukinumab or superior?
Yeah.
No.
Yeah. Is it superior to Secukinumab? One doctor is asking.
Yeah. So obviously there are studies that show that Secukinumab and this thing, Secukinumab is superior compared to Ustekinumab. There are some studies to show that, but and even the Ixekizumab, there are studies to show that. So, but still, see, everything, somebody will be superior, somebody will be inferior, but basically we have to say long-term disease control, efficacy, safety, and once in 3 months, that's the advantage of this. Obviously, time will tell us how good it is and how best you can use it in our day-to-day practice. So obviously there are these things.
Yeah. One doctor is asking, what are the chances of developing anti-drug antibodies?
Yeah, so that you can answer. What is considered
So
good?
see, as I can
Usually,
say,
the
yeah,
range is
yeah,
6% to 12%.
correct, correct. And but in our trial, none of these patients who were in the Ustekinumab group did not
Yeah.
develop any
So two patients in
immunogenicity.
Stellara group developed
Stellar, right?
anti-drug
Yeah, yeah.
antibodies, and
So that means
none in our Ustekinumab as such, at least in the study.
that means your biosimilar is better than the biologic.
We cannot accept that science yet, sir,
Yeah.
Yeah,
because
yeah.
there is a concept bio better, but that will take some 5 to 10 years more.
Right,
More.
right. Well, very interesting discussion. Time will tell us. We are very excited to use this molecule, this thing. Anybody wants to ask question to Dr. Nair for about this thing, Dr. Anchala, Dr. Rajiv, anything specific? Otherwise, last 2 minutes. Yeah. Well, thank you. There are no more questions. Thank you very much for all the support, and thank you, Reliance, for getting this molecule for Indian patients at the reasonable cost. And we are very happy to tell you the incident that I was just in Qatar last 2 days back. And when I told them that we get Omar Zubair at such a low cost and Tofacitinib at such a cost, they were actually really shocked that how can India get so much economical, less than $10-20 the cost of Tofacitinib. So that's the India. Basically, we make in India, made in India, and for India. So that's the advantage. So let's hope that this new molecule will give a new era in treating our patients and give best to the patients. This is from Kiran Godse, thanking you all for your support and this thing. And I hand it over to Reliance for the vote of thanks.
Thank you very much. And the key messages basically, as we all know, that it is world's first biosimilar, and in terms of safety, immunogenicity, and efficacy compared to that of the innovator, available in 45 and 90 mg PFS. And I very much thankful to Dr. Rajiv Shekri and Dr. Anchala for sparing their valuable time to be part of this webinar and giving all valuable inputs. Doctor, thank you once again, Dr. Rajiv and Dr. Anchala. And special thanks to Dr. Kiran Godse for moderating the entire webinar, and to all the doctors who have spent, in spite of being a Diwali night, everybody had come down and be part of this webinar. It is actually a great eye-opening for all of us, and we'll be definitely moving ahead with more webinars of such types, and maybe in more of the conferences. Thank you once again to all the doctors, and my
One
happy
small
Diwali. Yeah.
One small point, small
Yeah.
but important point, all doctors, we have already submitted this article of phase 3 for
Yes.
publication in the Indian
Yes.
Journal of Dermatology, Venereology and Nephrology, IJDVL.
Yeah.
We have a waiting period of at least, say, 2 months or so, and then the article will be published. Thank you
Yeah.
so much,
Yeah.
everyone.
Thank you very much.
Yeah.
Bye bye and happy Diwali.
And thank you very much. Happy and a
Happy
prosperous
Diwali to
Diwali
all.
to everyone and family. Thank you very much, doctors. Thank you. Thank you.
Thank you.